finally getting into the phase of muscle physiology where the muscle is actually moving, not just being excited. We're moving into the phase of cross-bridge cycling. Now, cross-bridge cycling is the physical movement of the muscle. But before we get to that, we have to look at what the muscle's like at low calcium levels. So basically, when the muscle's at rest. When the muscle's at rest, you'll notice troponin, this yellow right here. Troponin is blocking. So this site right here, this black site, is the binding site of myosin. Troponin is blocking that binding site by keeping tropomyosin in the way. So because troponin is in this shape right here, it's keeping tropomyosin, this structure, in place. So what that means is this whole line, which is tropomyosin, these orange lines, those are all blocking the binding sites of actin. That means you can't create a cross bridge. So what we need to do is somehow move that out of the way. But at low calcium levels, they're blocking. They're basically stopping the contraction. So then when the muscle finally goes through the whole process, we go through the initial stimulation from the action potential down across the sarcolemma, down the T-tubule. And we get to this point. The whole purpose is to increase calcium levels inside the muscle cell. So low calcium levels, troponin's in one shape, tropomyosin's blocking, you're not having contraction. But at higher intracellular calcium levels, so the calcium has been released from the terminal cisternae, from that sarcoplasmic reticulum. Now the increase in calcium, notice we have more calcium binding to troponin. Because we've increased the amount of calcium to bind, we're about ready to create some movement. So this additional calcium binds troponin, and when that additional calcium binds troponin, it allows for troponin, again this yellow, you can see how it's changing shape. It's going through what's called a conformational change. Now, a conformational change is a shape change. It's going to change its shape and pull tropomyosin out of the way. So you're noticing that the tropomyosin is being pulled back to expose the binding site on actin. So this increase in calcium allows for a conformational change of troponin, which then, by moving its shape, it pulls tropomyosin with it. As it pulls tropomyosin, it exposes the binding site on actin. That means the myosin head can now bind. So this is now the movement that allows for the mechanism to start. We have to go beyond this for the full idea of the cycling, but this is the initial phase that gets everything ready to go. So we've had the ability to bind. Now, this hasn't quite moved yet. This is just the binding. The actual cycling, which will be next, is what creates the movement. So that increase in calcium is what allowed the movement or the conformational change of troponin to pull tropomyosin away, exposing the binding site. So again, as a quick summary here of where we're at, we have the actual signal coming down. That's that neuromuscular junction. We're looking at how it gets across. Remember, calcium floods in, the acetylcholine goes across synaptic cleft. You bind to the actual motor end plates. Those are the acetylcholine receptors. You create a little bit of a gradient potential there. If that then builds up to reach threshold and action potential is formed, the action potential travels down the sarcolemma. That's the idea of action potential conduction or action potential propagation. It then heads down to the T-tubule. The signal, the action potential, goes down the T-tubule. As it goes down the T-tubule, it's going to stimulate the voltage-regulated calcium channels. Calcium is going to flood into the muscle cell. That calcium is then going to increase the concentration, which means it can bind to troponin. Binding with troponin, troponin goes through a conformational change, pulls tropomyosin out of the way, exposing the myosin binding site on actin. Myosin head binds, and we're ready for the cross-bridge cycling, the actual movement now. So that's what we're looking at. Think about it this way now. A little bit of a kind of a mini case study. Calcium is required for strong bones. We know that. We need calcium for our bones. But yet, whenever blood calcium levels fall, so your blood calcium levels decrease, your body's quick to pull calcium out of your bones. So what we know about contraction and everything, why would you be willing to sacrifice? So sacrifice bone density at this point. So what's the importance here? So why are you willing to sacrifice bone density to maintain blood calcium? Well, if you're thinking about that, we know calcium is used in 2 different points of this muscle physiology so far. We know calcium is used in the end of the motor neuron to allow the release of acetylcholine. We now know calcium is used here to bind troponin, to allow troponin to go through a shape change or conformational change to allow myosin to bind. If either one of those previous calcium steps didn't occur, didn't have enough calcium to go into the neuron, could you send acetylcholine across synaptic cleft, allowing the signal to reach the muscle? No. If you didn't have enough muscle calcium, could you release it to increase the shape change or conformational change of troponin? No. So calcium was vital for the bones for strength and density. You kind of need it in the nervous system and the muscular system as well. And while weaker bones won't necessarily kill you or cause a problem, they could cause problems eventually, but just a little bit here off, it's not going to cause a huge issue. You can always regenerate that. But if all of a sudden the muscle stimulus isn't working or the nervous stimulus isn't working, brain, heart can gotta be problematic there. So it's good to make sure you maintain homeostatic levels of blood calcium. Again, we're trying to tie the pieces together here as we keep working our way through the muscle chapter. So just about there, muscle physiology. We're at the point of just getting ready to move through the muscle itself.